today we have a very exciting speaker, Professor Jens Brüning, that is coming from Max Planck for metabolism research. He is the director of the Max Planck for metabolism research, which is in Germany. So he made all this way just to come and give us the talk and to meet with our teams. I'm especially excited about it because I think that his research has relevance to a lot of what we are doing, and it is connected to our community in ways that are so exciting and interesting, because some of the work that he's doing is pertaining to the mitochondrial fission proteins that were discovered here by Alex van der Blick, and now he's taking it to animal and to animal physiology and brain and in interaction with different organs. And so when we were sitting yesterday together, Alex and Jens with me together, I was looking at them and said, look, this person started with this protein, and look where this person took it. It's so amazing how our communities are expanding discoveries that are made from one lab to another lab in directions that we can never ever imagine. As I said before, whenever the metabolism theme is inviting a person to speak here, is because members of the metabolism theme have heard this person in a seminar and say this must be heard by the rest of our community. And this is exactly what I felt when I heard Professor Brüning giving a talk, and the way that he integrates metabolism with animal physiology and interaction between organs, and how our brain is connected to the metabolic organs. So somehow our community usually like to divide ourselves between those that are dealing with the organs that metabolize that metabolize the nutrients, and then organs that control what we eat, but not much in the interaction between them. And this is the beauty of Jens Brüning research. The list of the achievement of Jens Brüning is just so long, so it will take me until tomorrow. But I will tell you, I'll just pick from there. And so let's start. He started his doctorate at, he got his PhD in 1993. And that's exactly when mitochondria enter the eukaryote cell for the first time. And yes, and then he did his postdoctoral fellowship in Boston, and today he's the director of the Max Planck Institute for Metabolism Research. His research is mostly focused on the neuronal regulation of energy and glucose metabolism. And now he won some very important awards, and I would mention out of them those that most of us are familiar with, which is the Minkowski Award for the European Association for the Study of Diabetes. And then the Leibniz Award, the Leibniz Prize, which is one of the top awards in science that in the world, and specifically in Europe. I know it might embarrass you, but I'll say that what I usually get when I presented to the Leibniz Award, they said this is the pre-Nobel Prize Award. So one step before. Congratulations for that. That's amazing. So without further ado, please. Thank you.
Well, thanks a lot. Thanks, Orion, for this beautiful, exaggerating introduction. Let me try to see whether okay, cool. Now I need a pointer.
Share the screen as well.
Oh, sorry.
Yeah, do you mind if I?
First mistake.
You're totally fine. And your pointer, which is right down there.
Okay, cool. Well, it's a great pleasure really to be here in person. I think it's an amazing faculty that you have on board, and I mean, I hope you know how privileged you are to have really this broad interdisciplinary spectrum of outstanding colleagues. Some of whom I had the privilege to talk to this morning, and I'm really looking forward to the discussions because it's my first trip coming back to the States, you know, post-COVID, and so I haven't been to the US for 3 years and plus, and it's really inspiring, you know, to discuss from different aspects of metabolism, the research that is going on here. Okay, I want to give you a bit of a historical avenue, how I moved into the field. Because when I did, it all started out basically when I did my work as a postdoc in Ron Kahn's lab back in the early 90s. And so I'm trained as an clinical endocrinologist. And so I was always interested in trying to understand the mechanisms underlying insulin resistance during the development of type 2 diabetes. And so when I moved to Ron's lab, we decided to study how individual tissues contribute to the pleiotropic actions of insulin. And if you look at your biochemistry textbook, you will find a very easy picture that insulin released from the pancreas acts on insulin receptors in adipose tissue and skeletal muscle to promote glucose uptake and to act on insulin receptors being expressed on liver cells to suppress hepatic glucose production. And so I think this has been the dogma in the field for over decades. And that those are the three major metabolism regulatory insulin target tissues. However, what did strike us at the time was, if you really look carefully, or not even carefully, it's pretty much in your eyes, if you look where the insulin receptor is expressed, it's almost like a housekeeping gene. It's virtually expressed on all cell types of the organism. And the highest level you even find in the central nervous system, even higher levels of insulin receptor expression in some areas of the brain compared to what you find in liver, adipose tissue, and skeletal muscle. And so we really started out like more than 20 years ago trying to understand, you know, what is the physiological role of insulin action in those non-classical insulin target tissues. And on the other hand, then maybe getting some insights how insulin resistance could also act on those tissues to contribute to the pleiotropic syndrome of the metabolic syndrome and insulin resistance. And so we then, oops, sorry, I cannot move this here. Okay, there. And so we started out in a very naive approach, trying to, you know, start delineating the role of insulin signaling in the central nervous system, but generating mice which lacked the insulin receptor in the central nervous system, but retained expression of insulin signaling in peripheral target tissues. And so when I moved back to Cologne, we started generating those animals and characterizing them. And what we found was that there was not a major effect of removing insulin signaling from the brain on body weight regulation. There's a slight increase in body weight in those animals, pointing towards the possibility that also insulin acts as a feedback signal to communicate to the central nervous system how much energy is available in the periphery in form of glucose. But what was more striking is when we performed the simple insulin tolerance test in mice which lacked the insulin receptor in the brain, but retained insulin receptor expression in liver, muscle, and fat, that in response to exogenously applied insulin, the blood glucose lowering effect of insulin was attenuated when the mice lack insulin action in the central nervous system. So really indicating that insulin not only exclusively acts in the periphery to control systemic insulin sensitivity and glucose homeostasis, but in parallel or in addition, engages central nervous signaling. And so the question then, of course, was where and how in the brain is this encoded? And a very basic neurocircuitry, which has really gained a lot of interest over the last decades, is the melanocortin circuitry, which resides in the arcuate nucleus of the hypothalamus. And there you have many different neurons, and I will talk about more towards the end of the talk. But what we have learned is that there is a very highly conserved, very fundamental neurocircuitry residing in the arcuate nucleus, which is characterized by two neuronal populations, which are functionally antagonistic. So a subset of neurons, and there are only like 3,000 to 5,000 in mice and humans, express pro-opiomelanocortin, POMC, and those neurons are activated by leptin coming from the periphery or insulin, and those neurons then release alpha MSH, which acts on downstream neurons to stimulate the melanocortin-4 receptor to potently suppress feeding. So it's an anorexigenic food intake suppressing neurocircuitry, which is perfectly conserved between mice and humans. And mutations of POMC or the MC4 receptor lead to obesity, not only in mice, but also in humans. And right next to those neurons, there's a second population, also about 3,000 to 4,000 cells in mice and humans, which express another neuropeptide, which is called a GUDI-related peptide. And AGRP neurons are activated under starvation. They're inhibited by insulin and leptin. And when they are activated in starvation, they strongly promote feeding. And they in part act through the same melanocortin-4 receptor as AGRP acts as an inverse agonist for the MC4 receptor. So you have two functionally antagonistic neuronal populations: POMC neurons, food intake suppressing, AGRP neurons, food intake promoting, which respond to peripheral signals to adapt food intake and energy expenditure. So we then reason if those neurons are perfectly tailored to sense what is going on in terms of energy stores in the periphery of the organism, wouldn't it make sense that if they accordingly adapt their activity, that they not only then regulate how much energy you take up from outside of the body, but also how you partition energy across different organs? In other words, could it be that insulin acts on the same neurons, not only to control feeding, but also to control peripheral insulin sensitivity? And so some years ago, Christine Körner, a PhD student in my lab, then decided to knock out the insulin receptor, not globally from the central nervous system, but only either in those 3,000 food intake suppressing POMC neurons or in the food intake promoting AGRP neurons. And what she then performed were euglycemic hyperinsulinemic clamps, looking for the ability of insulin to suppress hepatic glucose production, because we had previously shown that in the brain-specific insulin receptor knockouts, insulin's ability to suppress hepatic glucose production was attenuated, pointing that the predominant regulation of peripheral glucose metabolism via the central nervous system through insulin is conveyed through the regulation of hepatic glucose production in the liver. And so what Chrissy then found is that if she looks at the ability of insulin to suppress hepatic glucose production in a control animal, insulin almost completely suppressed hepatic glucose production by 90%. If you remove the insulin receptor from the 3,000 food intake suppressing POMC neurons, you get the similar suppression of hepatic glucose production. But if you only remove it from 3,000 AGRP neurons, insulin loses about half of its ability to suppress hepatic glucose production. So really indicating that at least the critical component of neuronal insulin action in control of peripheral insulin sensitivity is relayed via this very specific AGRP neuronal population. And we could subsequently show how insulin actually inhibits the activity of those neurons through PI3 kinase dependent KTP channel regulation. And these studies have been confirmed by many other labs, Mimo at Shirley, Christoph Büttner at Mount Sinai. They all come to the same conclusion and could completely reproduce that removing different components of the insulin signaling machinery from AGRP neurons would impair systemic insulin sensitivity. One of the limitations of those experimental approaches, in our view, was that although you achieve a high cell type selectivity, the conditional knockouts of the insulin receptor was present throughout development. So we restricted it locally, but not timely. And so the big question that remained open was, are those neurons actually able to acutely regulate peripheral glucose homeostasis? And in the last, I would say, 10, 15 years, modern molecular neuroscience has really revolutionized the ability how we can interrogate molecular defined neurocircuits by remotely controlling acutely the activity of molecular defined neuronal populations. So we can express certain channels, so-called dread channels, in a specific subset of neurons, which do not respond to any endogenous substance from the organism, but only to chemicals that we can apply from outside. And therefore, we can basically gain remote control over the activity of those neurons. And when we do this for AGAP neurons, we can remote control basically on a minute-to-minute basis, either optogenetically or chemogenetically, the activity of AGAP neurons and ask what is the physiological consequence of the acute regulation of those neurons. And this is exactly what Sophie Stekolorum, at the time a postdoc in my lab, did. And she chemogenetically or optogenetically, through light-activated channels, acutely activated AGAP neurons and could show that doing this for as short as 15 or 20 minutes would impair systemic glucose tolerance and insulin sensitivity. So really showing that the acute regulation of AGAP neurons not only leads to massive feeding if we activate those neurons, but that they also acutely can regulate peripheral glucose homeostasis. And what Chrissy had not assessed in previous studies, when we did it like in 2007, the AGAP neuron-specific insulin receptor knockouts, since brown adipose tissue really wasn't as appreciated as it is today, we had not analyzed glucose uptake into brown adipose tissue in 2007, but did it now in the acute experiment. And what Sophie could show is that the acute regulation of AGAP neurons not only regulates hepatic glucose production, but also insulin-stimulated glucose uptake into brown adipose tissue, but not white adipose tissue. Okay, so what this brings us to is really what fascinates us. And I think Orion kind of introduced that in the major concept of our work. Well, the way we think about this neuronal control is that we have highly specialized neurons which are optimized to sense the energy state of the organism. They have receptors for leptin originating from adipose tissue. They have receptors for insulin to sense insulin. They directly respond to changes in glucose or fatty acid. So you really have neurons which are sitting there as a perfect senses what is going on in the periphery of the organism, and which then accordingly adapt behavioral responses such as feeding, energy expenditure, but as I've exemplified to you, also hepatic glucose production, lipolysis, and work from many other groups has extended that work, that AGAP neurons also adapt innate immune responses, bone mineral density, and through ascending projections, higher cognitive functions. So the way we're looking at this is that we have a system which is capable of adapting almost the entire integrative physiology to the actual energy state of the organism. And this is what really fascinates us. And of course, that also poses the possibility that if you have a dysregulation in those neurocircuits, let's say in obesity or metabolic disease, that this could account for the manifestation of a whole plethora of consequences other than overeating or dysregulated glucose metabolism. And this is why we think it's really interesting to study the mechanisms underlying this neuronal regulation. However, throughout my introduction, if you want, I've always highlighted, and this is a somewhat aged review from Mike Schwartz, the hormonal regulation of those neurons. So as I've told you, they respond to insulin, they respond to leptin. They also respond to ghrelin, which is released from the stomach in response to starvation. For example, ghrelin is a potent activator of those AGAP neurons. So we've always looked at those neurocircuits as responding to humoral signals originating from the periphery to instruct the brain about the energy state of the organism. If that would be the only way of how this system operates, you would have a very clear prediction on the temporal regulation of those neurons, right? If it requires those hormonal changes, you would predict If you're in starvation, AGRP neurons are active, POMC neurons are silent. You would ingest food, it gets deposited, leptin levels rise, and then eventually AGRP neurons are shut off and POMC neurons are activated. However, again, through very recent developments, we can now monitor online in freely behaving mice the activity of neurons in vivo by using calcium sensors. What you can do, you can put a calcium sensitive GFP, for example, into AGRP neurons, implant a laser fiber, and then record constantly the calcium signal from those neurons in freely behaving mice. And there were 3 groups who really did this in a pioneering way. It's Brett Lowell's group, Zach Knight's group, and Mark Enderman's group at Harvard. And they came to the same conclusion against the dogma that I've been telling you before. If you have starved animals, record the activity of AGRP neurons, basically the AGRP neuron gets shut off and the POMC neurons gets turned on the moment the animal sees and smells the food, even before taking the first bite. So this really tells you that there is much more to this regulation than just this humoral feedback. So there's no way for leptin levels to change. There's not even a single calorie inside the organism when the neurocircuitry adapts already to a state that is predicted to occur in the postprandial state. So sensory food perception is sufficient to let's say, hijack that neurocircuitry. And the big question is, what could that be good for? And so now, in line with our philosophy that those neurons not only control feeding, but also peripheral tissue physiology, we asked, well, could it be that the transient regulation of AGRP and POMC neurons in response to sensory food perception could serve to prime peripheral tissue responses to the postprandial state? And this is work by Klaus Brandt, who was a postdoc in my lab at the time, and also an independent group leader in Copenhagen. And so first, Klaus repeated the experiment that was published by Zach, and just to show you the scheme, so he put GCAMP, the calcium-sensitive GFP, into those anorexigenic POMC neurons, which are expected to be activated after eating. And he used 3 conditions. These are fasted mice, and you see the calcium signal. The moment here, when the arrow is indicated, this is when he either presented food to the animals, or what we called caged food. He just took the food, wrapped it up in a wire mesh so the animal could see it, smell it, but not consume it. Or he dropped in a wood block, which kind of looked like a food pellet. And these are the calcium responses of the POMC neurons. So the moment you drop in the food, there's a clear activation of the POMC neurons, which is similarly strong when you just drop in the food, which is wrapped up in the wire mesh that the animal cannot consume it, but nothing happens if you drop in the wood block. So really confirming, and if you put in a calcium insensitive GFP, nothing changes. So the question was then, could, knowing that those neurocircuits impinge on liver physiology, could the sensory food perception dependent regulation of POMC neurons somehow relate to peripheral tissue function? So then Klaus performed an RNA-seq experiment from liver of animals which were either fasted, they have been refed, or they have been exposed to caged food. And basically what you see here is a number of genes changing over a period of time, 30, 61, 2 and 4 hours. And you see this classical hepatic postprandial response. So hundreds of genes, as well described, change their expression when the animals are consuming food. However, there was a minor transcriptional response of approximately 50 genes, which were transiently induced just when the animal could see and smell the food, but not consume it. And this we've reproduced over and over again. So the question was then, what are those genes who transiently changed their expression in liver upon the sheer sensory perception of food? And when Klaus then put those into a gene ontology network analysis, he realized that the vast majority of those genes were target genes of XBP, the ER stress response transcriptional regulator. And they largely overlapped with those being upregulated during natural feeding, because we think the sensory perception is a natural component of what happens during eating. And so this really brings us back to the classical regulation of the ER stress response. And the question is, I mean, what is the prototypic regulation of this? And I think this is, pioneering work from Peter Walter and many others, showing this, basically this unconventional splicing of XBP, which drives the ER transcriptional response, which is governed by the sensor IRE, which resides in the ER membrane, and which is usually kept in an inactive state by being bound to the molecular chaperone. If then unfolded proteins accumulate in the ER, the chaperone is sequestered from IRE1, leads to the activation, splicing of XBP, and the initiation of an ER stress response. So in a conventional way, the ER stress response is viewed as a cell-autonomous response, which senses the occurrence of unfolded proteins in the ER, let's say in the liver cell, to initiate then to increase protein folding capacity and restoration of ER homeostasis. And so the question was then, is XBP activity really change dependent on the neuronal activity? And what Klaus found when performing phosphoproteomic analysis, that the activation, the sensory food perception, activated mTOR signaling in the liver, as did the artificial optogenetic stimulation of AGRP neurons here. And this led to an increased splicing of XBP1 and an increased expression of XBP target genes. And this study has been published a few years ago, but it really points us to the concept that the sensory perception of food is sufficient to transiently activate those neurons, which through sympathetic nerve activity regulation can activate an ER stress response in a non-cell autonomous manner to prime ER homeostasis for the upcoming demand during the postprandial state when all the nutrients come in. And so this, I think, takes us to maybe a more differentiated view how those energy state sensing neurocircuitries are used during different phases of the transition from fasting to feeding by engaging different stimuli. And so what we've uncovered is somewhat an example how the sheer sensory perception of food to activate those neurons can be used to prime peripheral tissues for the adaptation that will be required once the nutrients come in. Then there is a second phase, which I'm not going to talk about. It's a whole evolving, developing field, is actually those signals originating from gut innervation, from vagal responses upon nutrient consumption. And then only in the late long-term phase, those homeostatic hormonal signals kick in. And I think there's a lot still to be understood. What is the contribution of sensory perception-dependent priming? What is the feedback originating from different components reaching the gut? Until then, the long-term homeostatic hormonal responses kick in, which probably really maintain then stable body weight in long time. And I think these are all still interesting aspects to look at in the future. The other question, which has been, I think, open in discussion, is how actually does a hormone like insulin reach those neurons? So the classical view is that due to the specific makeup of the hypothalamus, I probably have it on the next slide, you have this very specific anatomy that you have the median eminence down here. Here's the third ventricle, and this is the arcuate nucleus where those AGRP and POMC neurons reside. And the general idea is, in line with the thinking that those neurons are specialized to sense what is going on in the periphery, they should not be sealed by a very tight blood-brain barrier, right? They're kind of made as antennas to the outside world, to the periphery, to understand what is going on there. But still, there is a blood-brain barrier. And the question is, how can insulin, as an example, really reach those neurons effectively coming from the vessels to the neurons? And this brings us to the very specific makeup of the blood-brain barrier, specifically in this region and other circumventricular organs. And you have the vessels in the median eminence, and you have specialized radial glia cells, which are lining the third ventricle, which are called tenocytes. And these tenocytes have very specific characteristics. They send, on one side, processes to the vessels, here at the basal part, and the tenocytes lining the ventricle more apically, they send projections to the neurons into the parenchyma. And there's increasing evidence that the tenocytes can be gatekeepers of hormonal access to the neurons. And this is exactly what we wanted to study, and this is work by Marta. It was really outstanding PhD student who just graduated and moved on to Boston to work in Mark Anderman's lab. And what Marta wanted to investigate, whether it could be that insulin receptors on tenocytes could contribute to the transport of insulin from the periphery to, let's say, the AGRP neurons, which we've shown are critical regulators of metabolism in response to insulin. And so first she did a very simple experiment, which is shown here. She took control animals, injected them with insulin, and looked for phospho-AKT as a readout for insulin-stimulated signaling. And you can see as early as 5 and 10 minutes after peripheral insulin injection, tenocytes lining the ventricle here exhibited a strong signal of phospho-AKT signaling, really indicating that tenocytes themselves are insulin-sensitive cells. Then she devised a genetic approach to specifically knock out the insulin receptor from tenocytes. And if you perform the same experiment, you see that insulin fails largely to activate AKT signaling in tenocytes if you have selectively removed the insulin receptor from those glia cells. And what is also interesting, if you take animals, put them on a high-fat diet for a prolonged period of time, and they become insulin resistant in the periphery of the organism, also insulin fails under those conditions to activate AKT signaling in tenocytes, really indicating that tenocytes are also becoming insulin resistant under conditions of obesity. So, and this also leads, if you remove the insulin receptor from the tenocytes, if you now look for insulin signaling in the neurons in the arcuate nucleus, you can see in control animals, in lean control animals, insulin rapidly activates AKT signaling in the arcuate nucleus of the hypothalamus. However, if you have removed the insulin receptor from the tenocytes, the ability of insulin to activate signaling in the neurons is largely attenuated, as is also seen in high-fat diet-induced obesity. And so our primary assumption was really, could it be insulin transport, maybe via insulin receptor-mediated transcytosis, that insulin receptors take up insulin, pass it on to the neurons via tenocytes. What Marta also tested, whether you need insulin receptors in vascular endothelial cells of the blood-brain barrier for a proper insulin action in the arcuate nucleus. And so in addition to knocking out the insulin receptor in the tenocytes, she also generated mice which lack insulin signaling, specifically in blood-brain barrier vascular endothelial cells. However, this had absolutely no consequence on insulin's ability to regulate signaling in the adjacent neurons. So really indicating that the insulin receptor signaling in the tenocytes, but not the blood-brain barrier vascular endothelial cells, is a prerequisite for insulin transport and action in the arcuate nucleus. So really, to come specifically to the question of insulin transport, we then wanted to test whether insulin uptake is altered. And so we Marta injected control animals with fluorescently labeled insulin peripherally. And you can see rapid accumulation of the fluorescently labeled insulin in the tenocytes, as well as in the neighboring neurons. And if you have removed the insulin receptor from the tenocytes fail to accumulate the fluorescently labeled insulin, as well as the neighboring neurons. So this really led us to the concept that you need insulin receptors on the radial glia cells, tenocytes, for insulin uptake and transport to the neighboring neurons. And so what happens as a physiological response? If you remove the insulin receptor from the tenocytes, the animals mildly gain weight, as we have previously shown for brain insulin receptor knockouts. They present with systemic insulin resistance. And if we perform euglycemic hyperinsulinemic clamps, you see a largely reduced glucose infusion rate during the euglycemic hyperinsulinemic clamp, so confirming the insulin resistance. They have a largely impaired ability of insulin to suppress hepatic glucose production, and they have a reduction in insulin-stimulated glucose uptake into brown adipose tissue. So largely mimicking the phenotype of what happens if we have only removed the insulin receptor from the AGIP neuron. So really pointing that insulin signaling on the tenocyte is a critical prerequisite for insulin's ability to reach its its final target cells, those AGIP neurons. So this was all still compatible with the model that insulin receptors on the tenocytes could act in the process of transcytosis, right? Transporting insulin. But then Marta started to investigate the response to other hormones originating from the periphery to regulate AGIP neurons, for example. And I've told you that another important, physiologically important regulator of AGIP neurons is ghrelin, which is released during starvation from the stomach to act on AGIP neurons to promote their activity. So if you take a control animal, inject ghrelin, this potently induces feeding, as shown here. If you do the same experiment in an animal which lacks the insulin receptor in the tenocyte, ghrelin almost completely loses its ability to acutely stimulate feeding. So really indicating that the defect in neuronal sensing responding in the absence of insulin signaling in the tenocyte is not restricted to insulin signaling, but apparently also the ability of the neurons to also respond to other signals originating from the periphery. And then Marta designed an experiment which I think was technically challenging, what I'm really proud of, what she did there. So what she asked, she wanted to study the in vivo online regulation of AGIP neuron activity in the presence or absence of insulin signaling in the tenocyte. So the classical way to manipulate, for example, insulin receptor expression in the tenocytes is Cre-LoxP mediated recombination. So we have mice which have a LoxP flanked insulin receptor, and we can use Cre to knock out the insulin receptor from the tenocyte. But she also then wanted to monitor the activity of AGIP neurons. And so we designed another mouse model which uses a different recombinase, which does not interact with the LoxP site to not knock out the insulin receptor, to express the DRE recombinase in AGIP neurons. And then we use the DRE-dependent virus to express GCAMP in the AGIP neurons. So this allowed us ultimately to monitor online the activity of AGIP neurons, either in control animals or those lacking the insulin receptor in the tenocyte. And so these are now the in vivo calcium recordings from those AGIP neurons. These are fasted mice. If you refeed the animals, AGIP neurons in control animals rapidly suppress their activity, as has been shown by many different groups. But if you don't have the insulin receptor on the tenocytes, the ability of refeeding to suppress AGIP neuron activity is largely compromised. If you inject ghrelin, this activates AGIP neurons in control animals, but if you don't have the insulin receptor on the tenocytes, this signal is largely attenuated. If you give serotonin, that potently suppresses AGIP neuron activity. Again, the ability of serotonin to regulate AGIP neurons is blunted in insulin receptor knockouts in the tenocytes, as is a response to CCK. So what this really means is that it's not only insulin receptors on tenocytes passing on insulin, but somehow setting a tone on the AGIP neurons to respond in any direction, either stimulatory or inhibitory. So providing basically the tonic signal of how AGIP neurons can flexibly adjust to changes in metabolism. And so the question is really, what could be such signals? And unfortunately, I don't have the answer to that. But what Marta also did is she performed ribosome profiling of tenocytes in control animals, those lacking the insulin receptor, or high-fat diet fed animals. And so the question is, do we find a common translational signature in tenocytes in the state of insulin resistance, it either being genetically determined or nutritionally induced? And what she found is actually that there is quite some genes dysregulated, which indicate a mitochondrial stress response in tenocytes in insulin receptor knockout animals, as well as high-fat diet fed animals. So the way we're thinking about this is that there is a more fundamental change of the tenocyte biology occurring in the absence of the insulin signaling, which then generates a signal to rewire the reactability of HRP neurons. And we are still working towards trying to define what the pathways could be. I'm not sure with time here, but okay, just two more quick stories. So one way how we think about changes that occur in obesity or I think what many people in the field are doing is trying to understand what are the physiological regulatory principles of metabolism and how do those then change in obesity. In other words, we try to understand what does insulin normally do, and then we start looking, well, how does insulin signaling change in obesity? But so the classical view of the field has been, okay, let's try to understand how HRP neurons normally function, and then try to see what happens to HRP neurons in obesity. How POMC neurons normally function, what happens to POMC neurons in obesity. But Alex Yais, a postdoc in my group, he took a different approach, and he wanted to see what neurons get turned on during the transition from normal child feeding to high-fat diet feeding, not really zooming on the neurocircuitries that we knew about, but maybe trying to identify novel neurons that specifically get activated under disease conditions or upon consumption of high calorically dense food. And so he used a technology which is called phosphoribotrap profiling. And this is a technology which was developed by Zach Knight when he was still a postdoc in Jeff Friedman's lab at Rockefeller. And so we know that many signals that lead to neuronal activation lead to mTOR activation, and mTOR activation leads to S6 phosphorylation. So what you can do is you can basically compare two states, let's say fasted animal, refed animal. You want to understand which neurons get activated in refeeding. You propose that the increase in activity, let's say in the refeeding state, will lead to increased S6 phosphorylation in that neuronal population. And since S6 is a ribosomal protein, you take a hypothalamic extract, then precipitate with an anti-phospho S6 antibody, and you should enrich for ribosomes originating from the activated neuron. And then you subject those associated RNAs to sequencing and get the molecular signature in an unbiased manner of the activated cell type. And this is exactly what Alex employed, trying to identify novel neurocircuitries which are activated in high-fat diet feeding. So he compared mice which had been on a normal chow diet or switched to a high-fat diet for 3 days, and then performed a phosphoridosome profiling from the hypothalamus and asked what are the associated transcripts of activated cells. And he came up with a neuropeptide signature of PNOC, which is pre-pro-nociceptin. And this is a neuronal population which previously hadn't been extensively studied in the arcuate nucleus of the hypothalamus. So this work indicated that those PNOC expressing neurons get activated in high-fat diet feeding, and we could confirm this in electrophysiological recordings that you find increased firing of genetically identified PNOC neurons in animals which had been fed a high-fat diet for 3 days. So are those the same or different neurons from the ones which we knew before? So we asked the question, are those PNOC neurons which also sit in the arcuate nucleus? Are they POMC neurons? Are they HRP neurons? Are they tyrosine hydroxylase positive dopaminergic neurons? And there was very little overlap with any of the known neuropeptidergic populations in the arcuate nucleus. So we find a new cell type sitting in this critical feeding regulatory center, which is readily activated upon consumption of highly caloric dense food. And, excuse me, those neurons are largely GABAergic inhibitory. So the question is, how could they impinge on the normal neurocircuitry that we know in terms of feeding? And so we performed an experiment together with Henning Fenseler, who is now a group leader. He trained with Brett Lowell in Boston, and now he's a group leader at our institute. And since the PNOC neurons were sitting right next to those anorexigenic POMC neurons, we performed the following experiment. We genetically marked POMC neurons. We put the light activatable channel into the neighboring PNOC neurons. And then if you shine a light on the slice, you record from the POMC neuron. And if the stimulation of the PNOC neuron, for example, releases GABA, this leads to an immediately light evoked current on the POMC neurons. And this is what you see here. You shine light, you activate the PNOC neuron, and you see an inhibitory current on the POMC neurons. This really tells you they're synaptically connected, and that POMC neurons are inhibited by the diet-activated PNOC neurons. And so the question is, if we manipulate those neurons now artificially, if we optogenetically activate them, this promotes feeding. And if we ablate them, as shown here, so these are the PNOC neurons in the arcuate nucleus sitting intermingled with the food intake suppressing POMC neurons. If we abrogate those neurons, this has no effect on body weight or food intake in normal chow diet fed animals, importantly. But if you switch the animals to a high-fat diet, you largely attenuate the overeating initiated by the high-fat diet feeding. So really indicating that those PNOC neurons are a new class of neurons which can confer diet-induced inhibition of the physiological anorexigenic POMC neurons. And I would like to close for the last 2 minutes to just with a study that we published a few months ago, in which I'm really also very, very happy about, because we wanted to do this for the longest time, and it just didn't have the right person to do this work. Because a lot of our understanding on the heterogeneity of cell types has been originating through the power of single cell sequencing in genomics. And there are quite some data sets out there of hypothalamic neuron single cell or single nuclear sequencing. The problem, in my view, is that every investigator does its own data set, does its own clustering, and then you come up with a nomenclature of the distinct studies from distinct subregions, let's say calling a POMC neuron cluster A25 something, the next study will call it something else. And so there's really no unified nomenclature of the different hypothalamic subtypes. And of course, you have a huge batch effect between different experimental conditions. And so what we wanted to do is to unify the nomenclature of hypothalamic cell types by trying to bioinformatically integrate the different data sets that are out there. And this is work which was really pioneered by Lukas Steuernagel, amazing bioinformatician in our group. And so basically the task is, relatively straightforward. Let's say you have two cell types which are the same, let's say AGRP neurons in one data set and the other data set. So you want to correct them to move the AGRP neurons together, but you don't want to move the AGRP neurons to the POMC neurons. You don't want to overcorrect to lose the granularity of the biology. And so what Lukas really went into is to systematically comparing different bioinformatic tools, asking as to how efficiently he could mix the different samples, but retaining the biological specificity, and going through thousands of permutations of settings in those, he really optimized the way to find the best approach for hypothalamic cells, to mix the cells the best from the different experimental conditions, but to retain the biological difference. And this is a machine learning based approach. It's called SCVI. But this is published also now. So basically, he then pulled out all the, at that time, available data sets. So there were like 17 different single cell sequencing data from the hypothalamus, also in part focusing on different regions of the hypothalamus. And then we also put in some new single nuclear sequencing data. And then he really managed to provide a reference map comprising about 500,000 cells from the arcuate nucleus, from the hypothalamus, and really clustering non-neuronal cell types out here and the neurons in blue. And so this really led us to, just published, I think, 2 months ago, now a unified reference map of different cell types in the hypothalamus, both non-neuronal as well as neuronal cell types. And this is built in a way that any additional study can be fed into the system, so that it's kind of a growing system. Like as more data sets are coming along, this can be more matured, but at least it keeps the path on a unified way. And then we could really use different modalities to confirm really the specificity of his predictions by in situ hybridization and different backtrap profiling experiments. What is also interesting, I think, is that single nuclear sequencing data very nicely integrated into this. And I think this is a big advantage for neuroscience and maybe also for peripheral tissues. If you have single cell sequencing approaches, you have to disintegrate the tissue, let's say with colony digest, which in itself, the whole process can change gene expression profiles so fundamentally that you want to snap freeze transcriptional states ideally. And that you can do with single nuclear sequencing. So let's say you want to look for fasting activated neurons, you just have a fat mouse, a fasted mouse, snap freeze the brain, and then process from the frozen brain the nuclei and subject them to single nuclear sequencing. So you get a much more accurate, timely frozen transcriptional profile. And so we could show that the single nuclear sequencing very well maps to the more classical single cell sequencing. And then when Lucas zoomed in, not only on FOS expression as a classical marker of cell activation, but he defined a core set of immediate early genes which indicated neuronal activation. Then using the single nuclear sequencing, he could now identify new cell types which respond to fasting. And sure enough, the top cell type that came up were AGRP neurons, which was very reassuring, but he came up with a complete new set of fasting activated neurons, which we're currently functionally investigating further. And with this, I will probably skip the last 5 slides and go with the summary, which is a bit lengthy. But I hope I've convinced you that the melanocortin neurons, so AGRP and POMC neurons, coordinate the adaptation of integrative physiology to the energy state of the organism. AGRP neurons mediate insulin's ability to regulate hepatic glucose production and brown adipose tissue glucose uptake through the central nervous system. In addition to the homeostatic hormonal feedback regulation, they integrate sensory information to prime anticipatory signals, as well as to receive gut vagal information. Sensory food perception primes organismal proteostasis via the melanocortin neurons. We think PNOC expressing neurons in the arcuate are rapidly activated upon consumption of highly palatable food, and we think they represent an attractive target for treatment of obesity. Insulin receptor expression in tenocytes is critical for insulin access and other hormones in the regulation of systemic insulin, glucose homeostasis. Obesity causes insulin resistance in tenocytes, and we think that HypoMap, this unified single-cell sequencing atlas, provides a unified platform for the functional interrogation of hypothalamic neurocircuits in metabolism. And with that, I really cordially thank you for the possibility of being with you. Looking forward to the continued discussions. I think I've managed the individuals who did the work in our lab. These are our funding sources, and we're always looking for talented postdocs. So if this speaks to any PhD student in the room, feel more free to apply to our group. Thank you very much for your attention.
Yes.
Any questions?
Yeah. So, okay, any questions? We're going to throw this thing on your head. Yeah.
Thank you. To go back to the last slide, you stated obesity causes insulin resistance in tannic cells.
Yes.
And perhaps you could state, because I didn't quite catch, were you giving us direction as to the mechanism of insulin resistance in those cells? Is it
No.
lack of the receptors?
No, it's the only, yes, I did not give you that indication. So we know that in the high-fat diet, because we have the translational profiles also from the high-fat diet fat and from the tennosites of the high-fat diet fat animals, and insulin receptor expression does not change, but yet insulin fails to activate signaling. So it really seems to be more post translational effect, as also seen in peripheral tissues. But we have some preliminary data indicating that it also might be lipotoxicity occurring in tennosites, which we think is largely single lipid driven, but this is what we're currently trying to investigate. We think that also excessive ceramide accumulation in the tennosite may contribute to insulin resistance, as we know from other peripheral tissues such as liver. But this is, ongoing studies.
So do you think that the metabolism, or let's say fat released from the liver in particles or not, can affect those tennosites?
Yes, that is basically our percent. Yes. I mean, we getting more and more data that it's ectopic ceramide accumulation also in tennosites, which may contribute to insulin resistance.
Okay, and do you see as part of the early stage of the cephalic phase of do you see already changes in the liver that would affect the tennosites?
No, this is some actually that we haven't specifically tested in the tennosite knockouts, which we still want to do. But what is really interesting, we've tested the cephalic phase response in high-fat diet fat animals, and that seems not to be impaired. So that is really interesting, that the immediate response that of the POMC neurons seems to be retained in high-fat diet fat animals. And we're still trying to understand, that seems to deteriorate with aging. This is what we find, that if you put animals for 12 weeks on a high-fat diet, that cephalic response is still maintained. But if you look in aged mice, let's say 1.5, 2 years, then it really deteriorates. And so that is not as straightforward. But with respect to the tennosites, it's also interesting that they predominantly affect AGFP, but not POMC neurons. And there's even a structural basis for this. There's beautiful work from Fanny Longley. She was a postdoc in Mimo Cheli's lab. Now she's in Lausanne. She has done very nice EM studies trying to see what are the specific neurons which are targeted by the tennosite processes. And she finds a very strong, even anatomical preference for making contacts to AGFP, not to POMC neurons. So that kind of makes sense that even under high-fat diet conditions, where the tennosites become insulin resistant, the POMC neurons still seem to be able to respond to the sensory perception.
Nice. Questions? Slavika?
So, given that some neurodegenerative diseases have been linked to diabetes, do we know whether this particular region is affected during neurodegeneration?
I think it's a great question. I think it's also, the discussion always goes both ways, right? I mean, so, people argue that diabetes, you might be promoting Alzheimer's disease, which, I mean, I think the clinical data are mixed out there, and the experimental data as well, to my personal view. So I mean, there's this whole idea about insulin resistance in the brain promoting Alzheimer's disease, which I don't buy into, honestly. But the other way around, there is evidence for dysfunctional AGFP and POMC neurons in AD models. And so much data is out there, but it's only a few studies, which really indicate that also neurodegeneration may affect those neurocircuitries, but this is as much as I'm aware of.
Yeah, and then actually that reminds me that there was a paper a few years ago that was showing that the best, this was looking at a very large population, a couple of million people, and this was looking at what is the best predictor of diabetes at age 40. If you're looking at different kind of tests that were run at age 16. And the best predictor was the IQ test. At age 16 was the best predictor of diabetes at age 40. And this
Which may be multifactorial, right?
Yes, which was really incredible.
But it highlights the role of the brain. I totally agree on.
Yeah, it's very interesting because they controlled for socioeconomical, etc. And diet, etc. And it was in very large group. More questions. Before I want to switch the questions to the direction of cancer, because we have a bunch of cancer research people here, and I was wondering so you're talking about metabolism changing the way our brain is thinking about food. Could metabolism in cancer somewhere in the body can then influence our brain dealing with metabolism, and can that maybe contribute to cachexia?
So this is exactly, I mean, that's the whole idea, is trying to understand the neuronal basis of cancer cachexia, for example. I mean, also to hijack that for neotherapies. But I think what I'm thinking, if I would be a cancer researcher and would keep with that concept of the integrative regulation of the periphery through the brain, and knowing that, you know, autonomic innervation can change innate and adaptive immune responses. And I think it's an emerging field, right? But that also the brain could control, at the end, cancer progression, metastasis through autonomic control of both cancer cell metabolism as well as innate immune responses. So I think what should be looked at is bidirectional communication. I mean, how can cancer impact on metabolism through the central nervous system, like in cachexia? But the other question, what I would be interested in, is also could maybe dysregulation of brain pathways with altered autonomic innervation also lead to changes in cancer progression and development.
Interesting. Slavika.
Reminded me, one of the historical perspective of aging was the metabolic, so energy expenditure is inversely correlated with, for example, in Drosophila model with lifespan in Drosophila. So given that this is such an important brain region regulating metabolic expenditure, would you think that it can be more than we anticipate, probably?
Absolutely. I mean, this is why what really fascinates us. So, I mean, and I think this whole proteostasis regulation, right? I mean, if you can regulate, I mean, protein quality control is a hallmark of aging, right? So being able to prime an appropriate protein folding capacity in liver, or to regulate autophagy. I mean, these are all key pathways in aging. And I mean, caloric restriction, at least, is a very well-defined way to extend lifespan or health span, at least in model organisms. A bit more debated than non-human primates, I guess, right? But I think this is the whole concept that we're really thinking about, is that you have those energy state sensing neurons, and by integration of integrated physiology, I think you will ultimately impinge also on age-related diseases in a broader sense than just metabolic disease.
Other questions? Okay, if not, thank you.